gonna start recording. Alright, so as we said, the bacterial DNA is found in the nucleoid region and in the plasmid region. You guys remember these steps? We go from replication. What comes after replication? Transcription. And then finally, what comes after that? Translation, right? This is how information flows. Okay, information flows that way: replication, transcription, translation. Okay. Remember that bacteria divides by binary fission, which creates two identical daughter cells. Okay, so this type of passing on DNA from the parent to the daughter cells, this type of transmission is called vertical transmission, and this goes from the parent to the daughter cells. This is called vertical transmission. Okay, from parent generation to the next generation. Now, bacteria also have another way through which they transfer DNA. The other way through which they transfer DNA is called horizontal transmission, horizontal gene transmission. Okay, and that's basically what we are gonna focus on in chapter 8. Okay, horizontal gene transmission. That's not my TV. Okay, so what are the ways or the mechanisms by which genes can be transferred horizontally? One way is transformation, another transduction, and then we have conjugation. 3 mechanisms. Transformation is basically where naked pieces of DNA is taken up by bacteria. So imagine in the environment, bacteria dies. When bacteria dies, it releases DNA into the environment. Nearby bacteria that sees that DNA takes it up and transforms it. The other one is transduction. Transduction requires a virus that basically takes DNA from one bacteria and transforms it into another bacteria. And then finally, we have conjugation. This one requires formation of a bridge between two bacteria. That bridge is called a pili or pilus, by which DNA gets transferred. Okay? Okay. So this is basically a introduction to the mechanisms. Now, as a result of these gene transfers, what happens is that bacteria acquire new features, new phenotypic features, new characteristics. Okay? New characteristics that allow it to become more, for example, resistant. For example, bacteria can acquire genes that allow it to become resistant to antibiotic treatments. This is what's called recombination, which is the recombination of the genes so that they acquire a new phenotypic characteristic or feature. Okay? So, recombination. Bacteria, as we know, have no exact equivalent mechanism to sexual reproduction. Okay? However, they can transfer and donate DNA from one bacteria to another. The end result of all this is formation of a new strain that is different from both the donor and from the original recipient. Okay? This will provide genes for resistance against antibiotics, for resistance against metabolic poisons, genes for new nutritional and metabolic capabilities, and genes for increased virulence and adaptation to the environment. Now, the word virulence, we're going to hear this a lot in this class from now on. What does the word virulence mean? The word virulence is referring to any structure that bacteria has which allow it to invade and cause infection. Any structure that bacteria has that allow it to invade and cause infection. For example, flagella can be a virulence factor. We call it a virulence factor. Okay? Capsule can be a virulence factor. Okay? Fimbriae, where bacteria can use it and attach and adhere to tissues. That's another virulence factor. So all of these virulence factors can be actually acquired through these genetic means. Okay? So recombinant. What's a recombinant?
Hi, I'm Lucy.
This is basically any organism that contains genes which originated from another organism. Okay? Any questions so far? Alright. So horizontal gene transfer in bacteria. Any transfer of DNA that results in organisms acquiring new genes that did not come from the parent organism, which is vertical transmission. Okay? So plasmids. Remember what plasmids are? Plasmids are small circular pieces of DNA. They contain their own origin of replication. Okay? The origin of replication is the site where replication starts. Plasmids replicate independently of the chromosome and the nucleoid. Remember, plasmids do not contain any genes that are required for survival. However, they do carry useful traits. Like what? Traits for antibiotic resistance, for example. Okay? Chromosome fragments. These chromosomal fragments must be integrated into the bacterial chromosome in order to be replicated and therefore passed on to the next daughter cells. Any questions so far? Okay, so let's